Thanks. Good morning. I'm Karen McDonald. I'm the PI of the CREATE IGERT at UC Davis, which combines plant science, molecular biology, and engineering. And I'll be talking about advances in the development of plants as molecular foundries. You've probably heard a lot about living foundries. There's been a lot of work done on microbial systems, and I hope to convince you that plants make excellent hosts for production of molecules. One reason is that plants have an unparalleled biosynthetic capabilities. They can produce very complex biomolecules that are used in a variety of applications, such as human therapeutics, as well as very complex enzymes with the ability to do post-translational modification of enzymes and proteins. Biotechnology has opened up even more possibilities for truly green biomanufacturing, and this just gives some examples of the different applications that people have been working on. One of the most recent ones is a milestone for the plant-made pharmaceuticals, in which is shown here. Just earlier this month, FDA approved the first plant cell culture-made human therapeutic for treatment of Gaucher's disease. But a variety of other applications making very complex molecules, biopolymers, biofortified corn, and even vaccines. So this outlines some of the plant biotechnology strategies, including stable integration of foreign DNA into the plant host nuclear genome to create stably transformed plants or plant cell suspension cultures, or insertion of the foreign DNA into the chloroplast genome. And so our IGERT trainees learn about all of these applications, in addition to the newer applications in which the foreign DNA is transiently expressed using either recombinant plant viruses or nature's genetic engineer known as Agrobacterium tumefaciens, and I'll talk a little bit more about that application. So transient expression using agrobacteria is a fairly well-studied process in which a piece of DNA on a plasmid in the agrobacteria is transferred into the plant cell, where transcription and translation and production of the recombinant protein occurs. The nice thing about this is that there's no need to generate transgenic plants. You get a very quick burst of gene expression and with a high protein yield, short production time on the scale of 1 week, and it's performed in a contained environment, and you can use both harvested plants as well as whole plants. The process involves a step called agroinfiltration, and this is just a step to bring the agrobacteria in close contact with the individual plant cells in the plant tissues. And basically the leaves or plant is submerged in a solution of the recombinant agrobacteria. A vacuum is applied, a moderate vacuum. The air from the interstitial spaces is removed from the leaf tissue. When the vacuum is released, the agrobacterial solution then infuses throughout the interstitial spaces, and the process that I just showed you takes place. So the idea here is really to use plants as a molecular foundry. And it would involve the steps of design, build, and test, typical engineering approaches, in which you might design a protein, an enzyme, or even multiple enzymes that would be used in a metabolic pathway. Then you'd use DNA building blocks, promoters, terminators. You might have tags to localize the protein to a specific place in the cell, and certainly the design of your gene of interest. Then you also need to decide on an agrobacterial strain. There's lots of different strains that can be used. You also have to decide on the plant host that you're going to use for this transient expression. So there's lots of opportunities for various combinations, virtually unlimitless. Then you have to build, and here the fact that the cost of synthetic DNA construction has gone down dramatically over the years is a real benefit. And you can actually synthesize many portions of this tDNA in the binary vector that is going to be inserted into the agrobacteria. And then you want to test. So you grow your plants, you grow your agrobacteria, perform the vacuum infiltration, then you incubate your plants for about a week, harvest the biomass, extract, purify. And these processes have actually been demonstrated at manufacturing scale. There are about 5 companies around the world that have produced large-scale facilities that allowed large-scale vacuum infiltration of either indoor-grown plants or greenhouse-grown plants, and basically following the same general principles that you see in biomanufacturing. So I'll just give you a couple of examples from our IGERT. This one is one of our students, Ben Lindenmuth, who's a former trainee. He's now at Bayer. And he looked at production of cellulase enzymes in plants to try to reduce the cost of biofuels. As you might realize, using the enzymatic route to produce biofuels, it's going to require very high quantities and low costs of those enzymes. Ben worked on producing a thermostable cellulase enzyme in tobacco plants and looked at the question of how does the expression of the full-length enzyme compare with just expressing the catalytic domain of the enzyme. So the process he followed is very similar. He designed the gene for his thermostable endoglucanase. He grew up tobacco. He vacuum infiltrated with the recombinant agrobacteria using harvested plants. He removed those harvested plants and then incubated them, extracted them, and measured the enzymatic activity. So he was able to show functional expression of both the full-length endoglucanase as well as just the catalytic domain over a 7-day time period, and this just shows the yields, milligrams per kilogram fresh weight, or yields in terms of the percent total soluble protein. Still fairly low. We need to work on increasing those expression levels. Another example is the work by Lucas Orzola, who worked on the production of an anthrax antitoxin for biodefense purposes. And the problem here is that there's a need for rapid, scalable, low-cost treatment for anthrax exposure. The specific research question he looked at is, can co-infiltration of agrobacteria containing plant gene silencing suppressors actually increase the productivity? This is the molecule that he was producing in tobacco plants again. It contains a site that mimics the anthrax receptor on the human cell, so it acts as a decoy so that it binds the protective antigen and prevents it from binding to the human cell. So this just shows the results from some of his work, which we've also published, but we looked at a variety of different gene silencing suppressors. So we would co-infiltrate with the agro containing the anthrax decoy protein, and also another agrobacteria containing these gene silencing suppressors. And one thing you can see is here's our control over here, this one, and with some of the gene silencing suppressors, particularly early on, this is 3.5 days post-infiltration, we could get very significant levels of our anthrax decoy protein. So with that, I'll stop and acknowledge the National Science Foundation, our partner, Tuskegee University, and our international partners as well as a lot of other contributors to our IGERT that contribute in a variety of different ways. So with that, thank you.